Hello everyone, the subject of today's lecture is gluconeogenesis, and we will also talk about the signaling pathways that put into place gluconeogenesis when it's appropriate and glycolysis when the opposite pathway is appropriate. So, just to remind you of where we are in chapter 13, today's subject is gluconeogenesis. We will talk about glycogen and the pentose pathway later on. So gluconeogenesis is essentially a reversal of glycolysis, and it's defined as the synthesis of glucose from non-carbohydrate precursors. So this is the subject for today's lecture. So in the where are we section, gluconeogenesis is here. So essentially we'll go from pyruvate to glucose. Remember, glycolysis was glucose to pyruvate, so we're going to go in the opposite direction. So pyruvate to glucose, that's gluconeogenesis. So how do we get to pyruvate? There are different ways we can get to pyruvate. And if you can get to pyruvate, you can get to glucose through gluconeogenesis. So if you have protein, for example, proteins can be broken down into amino acids. And when they're stripped of their nitrogens as NH3, we'll talk about that in a later chapter. If your carbon skeleton is a minimum of 3 carbons, you can convert that carbon skeleton to OAA or pyruvate or some other 3-carbon intermediate. So you have to have 3 carbons. 2 is not enough. So those can feed into either the converted to pyruvate or they might be able to be converted to something else along the pathway. For triacylglycerols, the triacylglycerol is broken down into the glycerol backbone, which has 3 carbons. That can be converted into dihydroxyacetone phosphate, and you can get to gluconeogenesis from there. But the bulk of the triacylglycerols can't, because fatty acids, we'll find out this in a later chapter, fatty acids are broken down to acetyl-CoA, and that only has 2 carbons. So again, if you have 3 or more carbons, you can get to gluconeogenesis, but 2 carbons is not enough. So there's no possible way, at least in mammals, no possible way to get from acetyl-CoA to glucose. That is impossible in mammals. So to put this into context, if we look over here on the right, say you're exercising and glycogen is broken down into glucose, and you break down the glucose to pyruvate to get 2 ATPs in glycolysis, and then the pyruvate's converted to lactate in the absence of oxygen. Lactate's a dead end. There isn't anything you can do with it. So this is described by the Cori cycle. So lactate leaves the muscle and then goes through the bloodstream and enters the liver. So the liver is able to carry out gluconeogenesis and converts the lactate back into pyruvate and then the pyruvate to glucose. This is an uphill pathway requiring ATP, so the breakdown of fatty acids can provide the ATP for that process. So another thing to note is that there are really 2 main tissues where gluconeogenesis takes place. The liver is one and the kidney is the other. So notice not the muscle. The muscle does not carry out gluconeogenesis. So I was sort of saying, well, gluconeogenesis is a reversal of glycolysis. Well, it isn't really, because if you remember from the glycolysis lecture that hexokinase, phosphofructokinase, and pyruvate kinase are all not reversible reactions. The rest of the reactions are, but not those 3. So that means if we're going to go from pyruvate up to glucose, which is shown here on the right-hand side. On the left-hand side, that's glycolysis. On the left-hand side is glycolysis. On the right-hand side is gluconeogenesis. We're going to have to find a different way to get past those enzymes. So let's talk about how we do that. So again, if gluconeogenesis is defined by pyruvate being converted to glucose, we can't go from pyruvate to phosphoenolpyruvate directly. That would be reversing the pyruvate kinase reaction, and we can't do that. And it actually takes us 2 reactions to do this. Stoichiometrically, remember that pyruvate's got 3 carbons, and we're going to glucose, which has 6. So we're going to need 2 pyruvates. So if you're going to keep track of the stoichiometry, you'd have to multiply everything here by 2. So a good exercise for you to do right now is to keep track of the number of ATPs or ATP equivalents that are required to carry out gluconeogenesis. So first, pyruvate is converted to OAA, and the enzyme here is pyruvate carboxylase. So carboxylase will take a bicarbonate and add a carboxylate group. So if you add a carboxylate group to pyruvate, you get OAA. Biotin is the B vitamin that you need because biotin is a carrier of bicarbonate. And this is an uphill reaction, so you're going to need ATP. So again, remember to multiply everything by 2. The next thing that happens is OAA is converted to phosphoenolpyruvate, and the CO2 is a leaving group, and we need GTP. That's an ATP equivalent, so we're going to count that as a phosphoanhydrate bond that we need to hydrolyze in order for this reaction to go. And what we do is we phosphorylate the OAA, we lose the CO2, and we end up with phosphoenolpyruvate. So we have successfully reversed the pyruvate kinase reaction, but we needed 2 other enzymes, and both of the enzymes required either an ATP or a GTP in order to make them happen. This is an interesting reaction. So if you the pyruvate carboxylase reaction, if you look down here at the bottom, takes place in the mitochondrial matrix. So if you look down here at the bottom, pyruvate is converted to OAA, and that's the pyruvate carboxylase reaction. And what actually happens next is OAA is converted to malate, and then malate leaves, and then malate goes back to OAA. And the reason we have to do that is because there's no shuttle to get OAA across. So we go from OAA to malate, malate leaves, malate goes back to OAA, and then here's the Pepsi-K reaction. The Pepsi-K reaction is in the cytosol. So OAA goes to phosphoenolpyruvate, and now we are in the cytosol, and we are on our way to continuing gluconeogenesis. So, again, let's look at the bottom. We've successfully converted the pyruvate to phosphoenolpyruvate. Now, all of these reactions are reversible. So, phosphoenolpyruvate is converted to 2-phosphoglycerate, and then isomerized to 3-phosphoglycerate, and we will need some ATP here to phosphorylate the 3-phosphoglycerate to 1,3-bisphosphoglycerate. And then NADH is needed. This is the dehydrogenase reaction that is also reversible, and we are going to convert the 1,3-bisphosphoglycerate to glyceraldehyde 3-phosphate. We'll lose one of the phosphate groups, so glyceraldehyde 3-phosphate is now more reduced. And again, everything here is going to multiply everything by 2, because once you get to the 2 glyceraldehyde 3-phosphates, one of them will be converted to dihydroxyacetone phosphate. And then you'll take 1 dihydroxyacetone phosphate and 1 glyceraldehyde 3-phosphate, and then carry out an aldol condensation and get to fructose 1,6-bisphosphate. So all of these reactions are completely reversible. So we would have to start with 2 phosphoenolpyruvates. All of these reactions will allow us to form 1 fructose 1,6-bisphosphate. But now we have to reverse the PFK reaction because we can't do that. So we were going to use a fructose 1,6-bisphosphatase. So here's the fructose 1,6-bisphosphate, and we're going to do a hydrolysis of the phosphate on the number 1 position. So a phosphatase enzyme is an enzyme that carries out the hydrolysis of a phosphate group. So fructose 1,6-bisphosphatase, we'll do a hydrolysis, and we'll go from fructose 1,6-bisphosphate to fructose 6-phosphate. Then the next step is fructose 6-phosphate is converted to glucose 6-phosphate, and that's phosphoglucose isomerase, and that's reversible, so that's not a problem. But now we get to glucose 6-phosphate, and we can't reverse the hexokinase reaction, so we'll need another enzyme, but it's another phosphatase because we're going from glucose 6-phosphate to glucose. And we will need glucose 6-phosphatase to carry out the hydrolysis of this phosphate group, and now we get glucose. So remember, gluconeogenesis occurs in the kidney and in the liver. And it's because those tissues express the enzymes that we need. So glucose 6-phosphatase is an enzyme that we need, and the liver and the kidney express the enzyme, and most other tissues don't. So that allows a phosphorylated, negatively charged phosphorylated sugar to be converted to glucose. And now glucose is neutral and can bind to transporters and can exit either the liver or the kidney, and then enters the bloodstream. And now other tissues can take up the glucose and use it for an energy source. there's another interesting tidbit here in terms of where these enzymes are found. The glucose 6-phosphatase enzyme is actually found in the ER. So the way that this reaction works is we have glucose 6-phosphate needs to be transported inside the lumen of the ER, and then the glucose 6-phosphatase is located here. It carries out the hydrolysis to glucose and phosphate, and then there are two separate transporters that transport the glucose and the phosphate into the cytosol, and then the glucose, once at the cytosol, can leave the cell. So if you took all of the reactions and added them all up, and again, we're starting with 2-pyruvate, and if you're keeping track, it costs 6 ATP equivalents to make this pathway exergonic. So 2 pyruvates, we needed 2 NADHs because glucose is more reduced than pyruvates, so we need an electron donor. So we need 2 NADHs, 4 ATPs, and 2 GTPs. So I would call that 6 ATP equivalents. So it is essentially a reversal of glycolysis, but glycolysis is exergonic. If gluconeogenesis is going to be exergonic, we're going to need to hydrolyze 6 phosphoanhydride bonds in order to make this pathway exergonic. Physiologically, what happens is if you would undergo a long-term fast, we store glycogen in the liver, and remember, the liver's job is to provide glucose homeostasis in the blood. So if you were to fast for a long period of time, liver glycogen is usually completely gone if you didn't eat by 24 hours. So gluconeogenesis is going to peak after that if you have a prolonged fast because liver glycogen would be depleted at this point. So again, all reactions have to be exergonic. The pathways in the cell don't happen unless they're exergonic. So if I look over here, if I start at the top with glucose, and I consider glycolysis, and I go from glucose to glucose 6-phosphate and then continue all the way to pyruvate, this will give me an exergonic pathway. In order to go from pyruvate to glucose, these reactions are exergonic as well, but it's because I had to spend 6 ATP in order to do that. So in the liver, in terms of when these pathways are active, during the fed state, glucose from the diet enters the liver and produces ATP and intermediates for biosynthesis. So after you've eaten, it's a good time for the liver to be carrying out biosynthetic reactions. So it'll need ATP to do that, and it will need the intermediates as well. Under the fasting state, though, glycolysis would be off and gluconeogenesis will be on, because if you're fasting, you don't have glucose from your diet, so you're going to need it from another source. So you'll take those non-carbohydrate precursors that we talked about at the beginning and convert those to glucose. So the main control point, as we learned in glycolysis, is PFK or PFK1. So the corresponding enzyme that is regulated is fructose 1,6-bisphosphatase. So the main way the pathway is regulated is if you want glycolysis to be on, all 3 of these enzymes, hexokinase, PFK1, and pyruvate kinase, they need to be operating. But if you want gluconeogenesis to be on during the fasted state, these enzymes all have to be turned off, and these enzymes have to be turned on. And again, the main, all of these enzymes are important, but these 2 are the main enzymes. So glycolysis will be on if PFK1 is active. Gluconeogenesis will be on if the fructose 1,6-bisphosphatase is active. And then, and these enzymes aren't both simultaneously active in general. If the fructose 1,6-bisphosphatase is active, PFK1 will be inactive, and if PFK1 is active, then fructose 1,6-bisphosphatase is inactive. So what we need to figure out when these pathways are active, and we want to keep memorization to a minimum if we can. But this is one thing you should memorize. Fructose 2,6-bisphosphate, which is not part of the pathway, is a major allosteric regulator of both glycolysis and gluconeogenesis. So this is something you're going to have to memorize. If you memorize this, you can figure everything else out. Fructose 2,6-bisphosphate stimulates PFK1, and it binds to the enzyme and encourages the R form or the active form. So fructose 2,6-bisphosphate will stimulate PFK1, glycolysis will be on, in the fed state in the liver. And it also inhibits the fructose 1,6-bisphosphatase. So gluconeogenesis will be off in the fed state, because in the fed state, you shouldn't be making glucose because you have glucose in your diet. So that's one way, by allosteric regulation. Another way is we're going to regulate by covalent modification. So protein kinase A is going to phosphorylate the tandem enzyme that catalyzes either the synthesis or the breakdown of the fructose 2,6-bisphosphate. And we've talked about this before, but we'll talk about it again now. So again, the reaction we're talking about is fructose 6-phosphate to fructose 2,6-bisphosphate. So what we do is we just take a little extra fructose 6-phosphate from our glycolysis and gluconeogenesis pathway, and we'll use that to make the fructose 2,6-bisphosphate. In order to do that, we'll need to phosphorylate the number 2 hydroxyl group, and we'll need PFK2 to do that. To carry out the reverse, we would use a phosphatase, and again, phosphatases catalyze the hydrolysis of a phosphate group, and that brings us back to fructose 6-phosphate. So remember that the way this is presented here, it's as if these are two separate enzymes, and they kind of look like that, but they're actually on one polypeptide chain. So if you see how this polypeptide chain folds, it kind of looks like there's two enzymes in one. It's like a tandem bicycle with two people. So this is one protein with two enzymes. So the yellow is the kinase. So this is the PFK2. So this is the active site where the fructose 6-phosphate would bind and the ATP would bind. And then this green, that's the phosphatase, and that's where the fructose 2,6-bisphosphate and the water would bind. And there's an N-terminus. I don't know exactly where it is in this figure, but it's somewhere near the kinase domain. So I don't exactly know where it is. It's probably around here somewhere. But there's a serine that can be phosphorylated, and that serine will determine which one of these enzymes is on and which is off. So when the kinase is on, the phosphatase is off, and when the phosphatase is on, the kinase is off. So in this tandem enzyme, these enzymes both aren't active at the same time. So let's talk about how we figure this out. I'm in the fed state. In the fed state, I want glycolysis to be on because I'm fed. I don't need to make glucose because I'm eating glucose. Glycolysis is on in the liver to provide ATP and biosynthetic intermediates. When you're fed, that's a good time to make biosynthetic compounds, glycogen and fat. So in this case, we want the PFK1 to be on. So that means we want to stimulate PFK1. And remember, fructose 2,6-bisphosphate is going to turn PFK1 on. That will turn glycolysis on. So we want fructose 2,6-bisphosphate to be there. And the way we make it is for the PFK2 to take a little fructose 6-phosphate, steal a little bit of that, and make the fructose 2,6-bisphosphate. So we want the PFK2 enzyme to be active. And once we have that, then the fructose 2,6-bisphosphate will bind to PFK1 and stimulate the enzyme. It will encourage the formation of the R form, and PFK1 will be on. And some of the fructose 2,6-bisphosphate will also bind to the fructose 1,6-bisphosphatase and encourage the formation of the T form and turn that off. I actually have a picture of this. This right here is the fructose 1,6-bisphosphatase. And here, where those little arrows are, there are 4, this is a tetramer, there are 4 molecules of fructose 2,6-bisphosphate that are binding, not to the active site, but somewhere other than the active site. The active site is actually here. And this is the T form. So that's where we have all these red arrows. So this is the T form. So when the concentration of fructose 2,6-bisphosphate is high, it is able to stimulate PFK1, inhibit fructose 1,6-bisphosphatase. That means glycolysis is on and gluconeogenesis is off. And when we're fed, we want glycolysis to be on to make ATP and intermediates for anabolic or building reactions. But we don't need gluconeogenesis to be on. We don't need to be making glucose because we're eating glucose. Okay, now how about in the fasted state? Well, in the fasted state, we want the opposite to take place. So in the fasted state, what we want is we want to get rid of the fructose 2,6-bisphosphate. So if we have the fructose 2,6-bisphosphate, now we want the fructose 2,6-bisphosphatase enzyme to be active to take off, to hydrolyze the phosphate on the number 2 position and make fructose 2,6-phosphate. We want the fructose 2,6-phosphate to be gone, and here's my animation. It's gone. So in the absence of fructose 2,6-bisphosphate, it's not there to stimulate PFK1, and it's also not there to inhibit gluconeogenesis. So glycolysis is off and gluconeogenesis is on because you're fasted, and gluconeogenesis needs to be on to provide glucose that you're not eating. So how does all of this work? This is all under hormonal control by hormones that are sensitive to blood glucose levels. So when you are fed, your pancreas has beta cells that will secrete insulin. They have a little glucose sensor. So when blood glucose is high, the beta cells of the islets of Langerhans in the pancreas will detect this, and insulin will be secreted. The opposite is true when blood glucose is low. When you're fasting, what happens is there are different cells, so the alpha cells of the pancreatic islets of Langerhans, and they will secrete glucagon. So when you're fed, your pancreas releases insulin and not glucagon. When you're fasted, your pancreas releases glucagon and not insulin. And this is what glucagon looks like. It's just a 29 amino acid peptide hormone, and that's what it looks like when you fold it up. So again, fed versus fasted. We're fed. When we are fed, anabolic pathways are active. And this is a good figure that shows this. It shows glycolysis, glucose to pyruvate, which makes 2,8-TP, as we talked about before. But notice how we can sort of siphon off a lot of these intermediates and make glycogen and lipids and nucleotides and lipids and more lipids and amino acids. We can make all of these things. And we want glycolysis to be on because when we're fed, this is a good time to be making all of these compounds because during the fed state, you have the resources to do that. So where I have the green arrows, all of these compounds can be siphoned off from glycolysis and be used to make these other compounds. But when we're fasted, there's the opposite. Catabolic pathways are active. Glucagon is released and gluconeogenesis is on to provide glucose that's not present in the diet. So what that means is the liver acts as a glucose buffer. So when dietary glucose is high, then the liver takes up glucose from the diet, from the portal vein, and then takes up maybe whatever it needs, and then releases the rest of that to the blood. But when blood glucose is low, the liver activates gluconeogenesis to release glucose into the bloodstream. So the liver's job is to maintain glucose homeostasis, and it happens using these insulin and glucagon hormones. So in general, the way hormones work, up here at the top, this is labeled endocrine cells. So the alpha and the beta cells of the islets of Langerhans and the pancreas that I was telling you about, those are the endocrine cells. And the endocrine cells will release hormones that travel through the bloodstream. And the target cells have to have receptors that can recognize these molecules. So if you look at the target cells here, the way this is drawn, this cell recognizes the green triangle, and this recognizes the red circle, and this recognizes the blue square. So not every target will respond to every signal that's in the blood. You have to have a receptor that's specific in order for you to do that. So how does this happen? Let's talk about how glucagon works. So here I am in the liver. This is the plasma membrane. Outside, this is in the bloodstream. So glucagon, so I have a little G right here, glucagon binds to a receptor. So this is a receptor. It's a transmembrane protein. It's shown here in this pink color right here. Actually, this is what it actually looks like right here down in the corner. Those are alpha helices. They kind of go back and forth and back and forth and back and forth. And this is the receptor. Glucagon binds to its receptor, and when it does, this is associated with a G protein. So these are called G protein coupled receptors because the receptors are coupled to the G protein. The G protein has 3 subunits, alpha, beta, and gamma, and when it's inactive, it has GDP bound. But when glucagon binds to the receptor, it interacts with the G protein, and the GDP is swapped for GTP. So GDP leaves and GTP hops right on. And you also jettison the beta and the gamma subunits. And then you activate an enzyme called adenylate cyclase. So again, so this G protein right here swaps GDP for GTP, dissociates from beta and gamma, and now you just have the alpha. So the G protein is active when you just have the alpha. That's these little lines right here are supposed to mean that it's active. And then it kind of cuddles up here to the adenylate cyclase and activates it. So the G protein activates adenylate cyclase, and what that does is that stimulates this enzyme, and adenylate cyclase takes intracellular ATP and catalyzes the conversion of ATP to cyclic AMP. So we call cyclic AMP the second messenger, because glucagon, you might notice, it never gets inside. It just binds to the receptor and then stuff happens, but the glucagon never gets inside. So we can consider glucagon to be the first messenger and cyclic AMP is the second messenger. Okay, so now what happens? Let me show you the reaction first. Here's ATP and here's the adenylate cyclase. So what happens is pyrophosphate's the leaving group and you make this cyclic compound right here. So this is cyclic AMP and it is our second messenger. What does cyclic AMP do? Well, it activates a protein called protein kinase A. And this is kind of a goofy diagram, but this is really what it looks like. So the protein kinase A, there's actually 2 copies. They're shown in blue, and that C is for catalytic. The way that this works is that there's another protein right here, and R is regulatory, and that's shown in red. So this regulatory subunit kind of plugs up the active site so that the enzyme's not active. But what happens is 4 molecules of cyclic AMP, so 2 bind over here to one R subunit, 2 bind to the other, and that causes a conformational change that releases 2 copies of protein kinase A. So now protein kinase A is active. So what does protein kinase A do? Well, it does what all good kinases do. Protein kinases phosphorylate other proteins. So the protein kinase A has a lot of targets, and we'll talk about those as we continue the class. But for today, it's that tandem enzyme that I was telling you about, that serine that was on the N-terminus. So remember, we have the PFK2 and the fructose 2,6-bisphosphatase. They have one protein chain. It's a tandem enzyme. So when the tandem enzyme is phosphorylated. So let's go over to the tandem enzyme again. Here's the kinase that's shown in yellow. So there's the kinase. So that's PFK2. And here's the phosphatase right here, which is shown in green. So what we've done is that serine right here, protein kinase A put a phosphate group on that. And when there's a phosphate group there, that turns the phosphatase on and the kinase off. So I have the phosphatase shown in green and the kinase shown in red because we are fasted and we want to get rid of fructose 2,6-bisphosphate. And the way we get rid of it is the fructose 2,6-bisphosphatase will hydrolyze that phosphate from the number 2 position, and now you just have regular old fructose 6-phosphate. And now the fructose 2,6-bisphosphate is gone. And it's not there to stimulate PFK1. It's not there to inhibit the fructose 1,6-bisphosphatase. So that means if the fructose 2,6-bisphosphate is not there, then glycolysis is off and gluconeogenesis is on. And gluconeogenesis is on because the fructose 2,6-bisphosphate would have inhibited it, but it's not there because we stimulated the enzyme that destroyed the fructose 2,6-bisphosphate. So gluconeogenesis is on, glycolysis is off. This is what we want during the fasted state, and glucagon is the hormone of the fasted state. So to put this sort of in context, in the fasted state, you're not eating, glucagon is released from the pancreas. The pancreas senses that blood glucose concentrations are low. Glucagon binds to its receptor, not all the details shown here, but activates the G protein, and the G protein activates adenylate cyclase. Adenylate cyclase takes ATP and converts it to cyclic AMP. That's the second messenger. 4 molecules of the cyclic AMP bind to protein kinase A and activate it. And then protein kinase A does what kinases do. It phosphorylates the tandem enzyme. And when there's a phosphate on that number 32 serine I was telling you about, the kinase is off, that's why it's shown in red, and the phosphatase is on. When the kinase is off and the phosphatase is on, fructose 2,6-bisphosphate is converted to fructose 6-phosphate. So if I go over here in my sequence of events, blood glucose level is low, you're not eating. Glucagon is secreted from the pancreas. This pathway shows how cyclic AMP concentration, the second messenger concentration, increases. Protein kinase A is activated by cyclic AMP, and one of the protein targets of protein kinase A is the tandem enzyme. The tandem enzyme, when it's phosphorylated, will inactivate PFK2, activate fructose 2,6-bisphosphatase. That means that fructose 2,6-bisphosphate will go down. That means PFK1 is inhibited. Fructose 1,6-bisphosphatase is activated, and gluconeogenesis increases. And that's what we want. Fasted, glucagon, more gluconeogenesis. So what about in the fed state? In the fed state, we don't secrete glucagon. We secrete insulin. And I'm not going to go over the mechanism of that because it's a little bit more complicated and it's beyond the scope of this course. But this is what I want you to know about insulin. A couple of things. Well, one of them is insulin activates an enzyme called protein phosphatase 1. So if glucagon activates protein kinase A, a kinase that phosphorylates enzymes, insulin is going to do the opposite. Insulin activates protein phosphatase 1. Protein phosphatase 1 will take the phosphates off. So this is what happens. This serine right here, with the serine number 32 right here, I'm going to take off the phosphate. There we go. I took off the phosphate. So when I take the phosphate off, which is what happens when in the fed state, that's insulin. You're going to take off insulin's going to activate protein phosphatase 1. Phosphatases take off phosphates by hydrolysis. Now the PFK2 is active and the fructose 2,6-bisphosphate is active. So again, going here with my tandem enzyme, the kinase is on and the phosphatase is off. So the kinase is on. That means we're going to take some fructose 6-phosphate and make fructose 2,6-bisphosphate. So now it's there. And the fructose 2,6-bisphosphate will stimulate PFK1 and inhibit fructose 1,6-bisphosphatase. Remember, we're fed. We want glycolysis to be on to have those anabolic pathways. We don't need gluconeogenesis to be on because you're eating. So you don't need to make glucose because you're eating glucose. So now the fructose 2,6-bisphosphate is stimulating glycolysis and inhibiting gluconeogenesis. So I didn't find a diagram in textbooks, so I made my own. Okay, so blood glucose concentration is high. That it increases the concentration of insulin being secreted from the pancreas. The PP1, protein phosphatase 1, is activated. That means that phosphatases remove phosphate groups from the tandem enzyme. That is going to inactivate fructose 2,6-bisphosphatase and activate PFK2. The fructose 2,6-bisphosphate concentration will go up. The fructose 2,6-bisphosphate stimulates PFK1 and inhibits fructose 1,6-bisphosphatase, and glycolysis will be on. In the fed state, that's what we want in the liver. So that was our whirlwind tour through gluconeogenesis and how it's regulated. I hope that you found this helpful, and I will see you in class.